Afternoon, folks. This is Professor Davis here. So I'm putting together a lecture for you all for the time that I'm going to be out of town. Just picking up where we left off, we've talked a little bit about energy. I'd like to carry on that discussion, say a bit more about how energy works, and go from there into a little bit about how our bodies manage their energy resources best. Now, we've established what energy is. We know that energy is going to be either potential, that is stored energy of position that is available to do work, or it is going to be kinetic energy of motion, energy that is actually doing work. And we've established some of the rules by which energy operates, our laws of thermodynamics. Firstly, the law of conservation of energy, which says that energy cannot be created or destroyed, only changing forms. And secondly, the law of entropy, which tells us that the total disorder in any closed system is going to gradually increase over time. But I want to zoom in a little bit more and talk about the kinds of chemical reactions that happen in your cells and the energy requirements for those reactions. Now, every reaction requires energy one way or another. You can't make any particle interact with another particle without expending energy. The question is going to come down to one of whether or not the particle in question already has enough energy to power its intended activity. In other words, do we have to put energy into the system to make the reaction go, or can it proceed on its own? Now, what this is going to come down to is basically, is the total energy present at the beginning of the reaction enough to make the product that we need to form at the end of the reaction? And there's an equation in chemistry and physics that is sometimes used to represent this. Some of you have no doubt seen an equation like this at some point if you've taken a chemistry or physics class. This might be a little bit different than the version you saw in your chem class, but it's going to illustrate what we need to do here. All right. You all see that equation there? All right, so see the formula is G equals H minus TS. And this is our equation for calculating what is sometimes called the free energy of a given reaction. Free energy is the energy available in order to carry out chemical reactions. If we want our reaction to be able to proceed spontaneously, if we want that reaction to be able to just go, then your free energy value needs to be greater than 0. You need a positive amount of free energy if you want that reaction to proceed. In this equation, free energy is represented with the letter G. Now, to calculate how much free energy you've got available for a given chemical reaction, and don't worry, folks, I'm not going to ask you to do this on a test or anything. I'm just showing you basically what this is so that you know what this all means and how it all works out. But if you want to know how much free energy is available for a given reaction, take the free energy value. Sorry, let's back up. Yeah, so we want to know the free energy value, G. First, you take the amount of energy that is available in the system when you start. That energy is called the enthalpy of the system. E-N-T-H-A-L-P-Y, enthalpy. So the enthalpy is the energy already present in the reactants when the reaction begins. Everything has some quantity of energy. You're not going to get away from that altogether. And so, yeah, that's your enthalpy, your starting value for energy, if you will. You're going to subtract from that the temperature at which the reaction operates multiplied by the entropy of the reaction. The entropy, of course, is the amount of energy we're going to lose to random disorder, to friction, to heat loss, to things like that. We multiply that by the temperature of the reaction, because the higher the temperature at which the reaction is taking place, the more energy is going to be shed in the form of heat and thereby wasted. You guys all know that a machine operating at a high temperature is probably less efficient than one operating at a lower temperature. It's losing more of its energy in the form of heat. So in other words, if I want to know my total value for free energy, G, I take my starting amount of energy, H, the enthalpy, subtract from that the amount of entropy loss I can expect times the temperature at which the reaction is running. So, in other words, total I start with minus the costs I can expect to incur along the way. That tells me my value for free energy. By the way, a thing to know here, that when we're counting temperatures in this situation, we do not use the Celsius scale. We use a related scale that is particular to physics and chemistry called the Kelvin scale. Note that I say it is in kelvins, not in degrees Kelvin, because it's a it's not a relative scale like the Celsius scale is a relative scale. 0 Celsius is compared to the freezing point of water. An object at 0 Celsius still has energy. The Kelvin scale is an absolute scale measuring the total energy content of the object in question. 0 Kelvin would be a situation in which you literally had no energy at all. There's a name for that in physics. It's called absolute zero. That's the point in which an object has literally no energy. All molecular motion stops. In terms of temperature, 1 Kelvin is the same amount of change as 1 degree Celsius. If you want to know your temperature in kelvins, take your temperature in Celsius, add 273.15. 273.15. So in other words, if I am at 0 Celsius, that's actually 273.15 kelvins. And by extension, you know that absolute zero, the coldest possible temperature, a temperature at which something has no energy at all, that would be negative 273.15 degrees Celsius, or somewhere in the neighborhood of negative 461 Fahrenheit. So the Kelvin scale is used when it's important for us to know the absolute energy content of the object in question in chemistry or physics, not the relative temperature as compared to the freezing point of water. All right, so you did this bit of math here, right? You plugged in your value for enthalpy, your starting energy. You subtracted from that your entropy times the temperature in kelvins, and determined there what your value for free energy comes out to be. This is going to tell us whether or not our reaction is able to proceed spontaneously. Now, there are two possibilities for how this can go. Your reaction can be exothermic, or it can be endothermic. Alternate terms you could use there that mean the same thing. An exothermic reaction could also be called exergonic, E-X-E-R-G-O-N-I-C. An endothermic reaction could be called endergonic, E-N-D-E-R-G-O-N-I-C. By the way, if you want to follow along with vocabulary for this section, it might be worth your while to pause this. Make sure you have the study guide for exam 2 that's up on Blackboard. Keep that handy, because then you can just take a look at these terms as I say them and not have to worry about writing down the spellings quickly. Now, the term exothermic literally means to give off heat or to give off temperature. Endothermic means to take in heat or to take in temperature. The equivalent terms endergonic and exergonic mean to give off energy for exergonic or to take in energy for endergonic. I don't care which one you guys use, but more often in chemistry, you tend to hear exothermic and endothermic. Now, if you do your math on this and you find out that your value for G is positive, in other words, the amount of energy you're going to lose along the way is less than the amount of energy you had to start with, your reaction is going to be an exothermic reaction. So an exothermic reaction is one in which the final value for free energy is greater than 0. In an exothermic reaction, you can have the reaction proceed spontaneously. It can go without you having to add energy to make it happen. Passive transport is exothermic. Now, the thing about this is that an exothermic reaction is actually emitting energy. The term exothermic, to give off heat. During an exothermic reaction, you are shedding heat or light, or you're otherwise expending energy in some fashion. Your products are going to have less energy than your reactants did. You had an amount of energy in the reactants, your enthalpy. You subtracted some from that along the way. You have some left, but ultimately your final products of the reaction are going to have less energy than the reactants had going in, because you've lost some during the way. You've shed some energy. It's exothermic. You've given off some heat or other energy. Now let us just suppose you do your math here, and your reaction comes out such that the value for G is equal to or less than 0. In other words, you have no free energy in the course of all this. The amount you're going to lose along the way to entropy is going to exceed or equal what you've got to work with. If that is the case, then your reaction cannot proceed spontaneously. You don't have any free energy to make that happen. Therefore, if your value for G is equal to or less than 0, we would say your reaction is an endothermic reaction. Now, an endothermic reaction cannot proceed spontaneously because you don't have energy to activate it. Doesn't mean it can't happen. The only way to make an endothermic reaction go is to add energy to the system. You're going to have to heat something up, shine a light on it, shake it up, do something to add energy that is not presently there. Once you put in the energy, you can get the value for G above 0, and you can actually make that reaction proceed. So an endothermic reaction, take that word endothermic, to take in heat or to take in energy. Endothermic reactions require you to put energy into the system to make the reaction go. Now, because you're adding energy, what this is going to mean is that the products at the end of the reaction are going to have more energy than the reactants did going in. That your final energy value for the reactants is going to be less than the final energy value of the products you get out of the system. And the only way that can happen is if you added energy, since after all, the first law tells us energy cannot be created or destroyed. I couldn't create more energy. So if I have more energy at the end than I had at the beginning, you know the reaction had to be endothermic. We had to get energy coming into the system from somewhere outside. Now, what this often means in practical terms, it's not always the case, but it's often the case, is you will find that exothermic reactions tend to be energy-releasing reactions. Something moves down a gradient and releases energy along the way, for example, like you saw salt ions doing in coupled transport to move glucose. Or you might find that you are releasing energy from bonds that can be used elsewhere. Like, for example, you digest your food. You break the chemical bonds in glucose. That is an exothermic reaction. You're releasing energy in stored form, which can then be used by the body to do other things. Some of that energy is inevitably lost in the form of heat radiation, and as such, you don't get every joule of energy out of that chemical bond or every calorie of energy out of it that was theoretically stored there. So my exothermic reaction is an energy-releasing reaction. You will find that often, but not always, exothermic reactions are catabolic. C-A-T-A-B-O-L-I-C. Catabolic. A catabolic reaction is one in which chemical bonds are broken, very often then releasing energy along the way. You might form new chemical bonds at the end, but they're going to be lower energy bonds than the ones you started. By contrast, you will find that your endothermic reactions are very often anabolic. A-N-A-B-O-L-I-C. Anabolic. An anabolic reaction is going to be a chemical building reaction. It's one in which we're forming new chemical bonds, that the bonds we have at the end are stronger than the ones we had at the beginning. They have more energy stored in them. So there's not a perfect correspondence between catabolic and exothermic and anabolic and endothermic, but it's often a close association. That catabolic reactions tend to be exothermic, they release energy. Anabolic reactions tend to be endothermic, they store energy. All right, making sense? All right, so keeping that in mind, we now know that your chemical reactions in your body can happen in a couple different ways. Now, one of these is not better than the other one. One of them is not more necessary than the other one. They're doing different things. Anytime your body is building new molecules, it's forming new chemical bonds, whether that means synthesizing enzymes, healing from a wound, growth, whatever. You're going to need to build new chemical bonds. You're going to have to spend energy to do that. You're going to be engaging in anabolic reactions that are probably endothermic, that are taking in energy. By contrast, when your body digests stuff, when you break down your food and so forth, you want to be releasing energy at that time. So a reaction like digestion is going to be a catabolic reaction, in which chemical bonds are broken and energy is released. It's going to be exothermic. You need both of those things going on, depending on the kind of task that you're biologically carrying out at the time. So, pardon me, folks. All right, now important thing to consider here. I told you that if you have a free energy value that is greater than 0, your reaction can proceed spontaneously. Okay, that's good. But spontaneous does not necessarily mean fast. Sorry, my camera's at a funny angle here. And I gesture too much. I need to have you be able to see my hands when I do that. But anyhow, though, spontaneous does not mean fast. Y'all ever sit outside on a warm summer night with nothing better to do and watch rust forming on the bumper of your car? Of course you haven't done that. For one thing, your life is more interesting than that. And secondly, it would be too slow. The formation of rust is technically an exothermic reaction, but it's also one that proceeds really, really slowly. Just because something has enough energy to happen spontaneously does not mean it has enough energy to happen quickly. And that's really important. Because we might supply enough energy to make a reaction go, but we may not have enough energy to make it go as quickly as we want it to. That reaction might not be rolling the way we need it. And in our body systems, we can't wait around. Your cells need to be able to do what they need to do now. They need to be able to break bonds now, to fight off demands from viruses or bacteria that are assaulting the cell now. We need to be able to respond to the environment as quickly as these things happen. Can't wait around all day for that to take place. For me to say these words to you, I have to have chemical reactions happening in my brain to put together the memories of these concepts with the memories of the particular muscle movements and tongue movements and so forth I need to engage in to make the noises that communicate these ideas. I have to have more chemical reactions that channel those nerve signals from my brain down to the nerves that control the muscles in my face and my jaws and my voice box and so forth. And still more chemical reactions to make those muscles contract and carry out the movements necessary for me to say all this. And that has to happen in the blink of an eye. So I have to have a reaction that is not only spontaneous, but fast. And sometimes if I only have a wee little bit of energy over my threshold, if I'm only a little bit over zero in my value for free energy, that may not go very quickly. You see, every reaction, endothermic or exothermic, needs a little bit of energy to get it tipped off. We call it the activation energy. Think of it as the first domino that you push to set off the rest of the chain. And all reactions require a little bit of activation energy. If your value for G is only a little tiny bit above zero, you may not have a lot of activation energy to spare. And that reaction, therefore, might only proceed very, very slowly. We technically have enough energy for it to happen on its own, but we may not have enough energy for it to happen as fast as we need it to. So what do? There are two basic solutions. One of them, you might call the easy solution, is to throw more energy into the system. Add more fuel to the fire, right? Heat things up, spend more energy, add energy, and you will make the reaction go faster. You'll hit that tipping point more quickly. Yeah, you can absolutely do that if you want to. But it's the worst way to make this happen. It's inefficient. It's forcing you as an organism to come up with more energy to get the same task done. And we as living things, in accordance with the second law of entropy, have to continuously replenish our energy so that we don't break down. And therefore, we want to use our available energy as efficiently as we possibly can. And throwing more power into the system to make a reaction start when there's technically already enough energy to make it go isn't an efficient way of doing that. So what do we do better? The thing we do better, folks, is that instead of trying to add more energy to speed up the reaction, living systems typically will try to lower the activation threshold. We will try to reduce the amount of energy needed to make the reaction start, lower the activation energy. That way, we can get the same work out of a smaller quantity of energy. We can use the energy we've got more efficiently. That's the name of the game. Imagine you've got a bunch of tasks you have to do today, right? You got to go out to the grocery store, and you got to go out to Target, and maybe you have to go to the car wash, and all these things need to be done. So you get in your car, and you drive to the car wash, then you drive home, then you go to the grocery store, and then you drive home, and then you go to Target, and then you drive home. Yeah, you can do it that way, but it's probably less efficient than if you only left your house once, made a loop, visited all those places, and then returned home. Getting the same work done more efficiently. In a lot of ways, that's what living cells try to do. Rather than throwing more energy onto the pile to make that reaction proceed faster, our cells endeavor to find a way to use the available energy as efficiently as they possibly can, getting more bang for our buck, getting more work out of the energy we've already got. In other words, lowering the activation threshold so that spontaneous reaction happens faster without having to add more energy to the system. Make sense? Now, how do we do that? If you're a chemist and you want to speed up a chemical reaction without heating it up more or adding more electricity or whatever else you would do, your way of doing that is going to be to add a catalyst. That's what catalysts are for, folks. A catalyst speeds up a chemical reaction without participating in the reaction. The catalyst doesn't add energy to the system. The catalyst does not combine with the reactants. It's kind of a helper in the reaction. It's not a new reactant. And a catalyst can't transform an endothermic reaction into an exothermic reaction. We can't get rid of the need to have energy in the first place. What our catalyst does is it helps us to make the best use of the energy we've got. It makes the reaction more efficient by guiding the energy in the right way, so that we get more work out of that same quantity of energy, and then thereby make that reaction happen faster. That's what catalysts are for. And in biology, you all know the name of our catalysts. Our catalysts are the molecules called enzymes. So that's ultimately where I'm going with this. Let's talk about enzymes. Enzymes are organic catalysts. They are the molecules that speed up the chemistry of your body enough to make it feasible, to make it so that I can say these words as fast as I am thinking them and have them get to you in a meaningful way. Now, an enzyme, just to give you an idea how much an enzyme can speed up a reaction. Let's talk about a common reaction that happens in your cells, well, not in your cells, but in your bloodstream all the time. You need carbonic acid in your bloodstream. Remember carbonic acid? It's one of the buffers that regulates the pH of your blood. You always need to be making carbonic acid. Now, the process of making carbonic acid is easy. It's an exothermic process. You combine a molecule of water with a molecule of carbon dioxide. This happens in our bloodstream all the time. Without any help, our bodies can make carbonic acid at a rate of about 200 molecules per hour. Sounds good? Nowhere near enough. We're going to need a lot more carbonic acid than that for the 5 or 5.5 liters of blood you have in your body. So let's throw in an enzyme. Let's help this reaction out with a catalyst called carbonic anhydrase. A-N-H-Y-D-R-A-S-E. Carbonic anhydrase. Carbonic anhydrase will speed up this reaction dramatically. In fact, it will speed it up so much that we'll go from a rate of 200 molecules of carbonic acid per hour to 600 molecules of carbonic acid per second. 3 times as much every single second as the body would by itself, make it an entire hour. That is the power of enzymes, folks. That's what a catalyst can do. It can greatly accelerate that reaction, and that's the kind of thing we need to make biological reactions go. It's that kind of acceleration that makes it possible for me to think a word and say that word right away, with a whole chain of chemical reactions between my brain and my tongue that have to happen for that to take place. So enzymes are crucial to maintaining the speed at which our chemistry goes. Most life processes that we depend on quite simply could not happen fast enough without their help. So how do they do it? How does an enzyme actually speed up a chemical reaction? Let me just show you a diagram here of an enzyme and tell you a little bit about some of its parts. All right, folks, so here is a terrible diagram of an enzyme. First off, look at the kind of blobby shape here. Enzymes are globular proteins. They don't have a specific defined shape. They are going to be highly variable in shape. They're flexible. They have to be because they have to be able to flex themselves around whatever substances they are helping to react. Any substance that an enzyme is helping to react, you might recall from our cheesemaking lab, is called its substrate. S-U-B-S-T-R-A-T-E, substrates. Now, my enzyme might be flexible, but it does have a couple of gaps or clefts in its surface of fixed shape. Take a look at the bottom of that enzyme here. You can see that we have a couple of gaps in the surface of the enzyme called the active sites, sometimes called activation sites. I'm okay either way. Your active sites are the places where the substrates will bind to the enzyme. Every active site has a specific shape that only fits the shape of that particular substrate. There are no all-purpose enzymes. Every enzyme helps to catalyze one particular kind of chemical reaction, and that reaction only. It does not have another job that it does. Its active sites only match that particular kind of material. Now, your enzyme always has active sites to match its substrates. Your enzyme may or may not have an allosteric site. An allosteric site is a binding site for a helper molecule. that might assist the enzyme with its activities or help control it. We'll talk about that in a few minutes here. And finally, my enzyme might have an ATP binding site. If the enzyme is helping with an endothermic reaction, remember enzymes can't make endothermic reactions into exothermic ones. All they can do is improve the energy efficiency of the reaction overall. So if the enzyme is one that's regulating an endothermic reaction, we still need to put energy into the system to make the reaction go. Therefore, endothermic enzymes very often are going to have... Is my camera blinking? I don't know what's going on there. All right. But anyhow, though, my endothermic enzymes are typically going to have an ATP binding site, a gap in the surface of the enzyme where a molecule of ATP can bind in place and be broken down and transfer energy to the reactants. So my enzyme is this globular protein, flexible shape, that has active sites to bind its substrates. It may or may not have an allosteric site for a helper molecule or a regulatory molecule. And it may or may not have an ATP binding site, depending on whether the reaction the enzyme helps with is endothermic or exothermic. Endothermic enzymes probably have an ATP binding site. Exothermic enzymes probably don't. Now, given that, enzymes can help with reactions in two basic ways, and ultimately it all depends on the question of whether or not the reaction in question is a catabolic one or an anabolic one. So a lot of people hear enzyme and they think, oh, that helps to break stuff down, that helps to digest stuff. But enzymes are just as likely to carry out an anabolic function, to help to build something. And they're going to help with reactions in two different ways. If my enzyme is helping to carry out a catabolic reaction, that is, it's a reaction in which we're trying to break two things apart, like a digestive enzyme, here's how it's going to work. First off, the substrate is going to bind to the enzyme. It's going to plug into the activation sites present on that enzyme surface. Once the substrate is bound to the surface of the enzyme is going to apply stress to the bond between them. Think about like how if you wanted to like weaken a piece of paper to make it easier to tear, you put a crease in it, right? And then you fold it a couple of times along that crease, fold it back and forth. You're weakening the bonds in the paper. You're disrupting it to make it easier to tear. That's what our enzymes do. For a catabolic enzyme, it is stressing the bond between two substrates, making that bond easier to break. As a result, those substrates separate from one another much more rapidly than they would have done on their own. Once the substrates have been pulled apart by the enzyme, they'll separate from the activation sites and go about their business. The cell has now digested that molecule, broken it down into smaller parts. The activation sites on the enzyme are now free to do that job again. The enzyme itself is a catalyst. It was not a participant in the reaction. It was not chemically changed in the process. So those activation sites are still there, still ready to plug in another copy of that molecule and carry out that same reaction all over again. Now, how about if our reaction in question is anabolic? Suppose that our goal is to bind two things together. The enzyme is going to start off the same way. Substrates are going to plug into its active sites. But in this case, once the substrates have bound to the active sites, the enzyme is going to move them together. It's basically going to force the substrates together so that they interact much more rapidly than they would have if they had to simply find each other randomly in the interior of the cell. And as a result of that, that reaction happens considerably faster than it would have happened on its own. That reaction is greatly accelerated because those substrates have been physically brought together. Once they have combined, they'll pull apart from the enzyme, leave the active sites behind. We now have this newly formed molecule. And then the enzyme is free to do that reaction again. As with a catabolic reaction, the enzyme itself is not changed by the reaction we carried out. The enzyme is ready, available, and good to carry out that reaction as many times as we keep supplying substrates to it. If the reaction in question required energy in either case, we would precede all of what I just described by binding a molecule of ATP to the enzyme. The enzyme would then break down the ATP, transfer the energy to the substrates, allowing them to react in the way that we need them to react, whether that's coming together or breaking apart. So fundamentally, enzymes have two basic ways of operating. For a catabolic reaction, the enzymes stress the bond between the substrates to make it easier to break down. For an anabolic reaction, the enzyme is going to bring the two substrates together and force them to interact with each other. In either case, the reaction is happening way faster than it would have if the substrates had to do this all by themselves in the cytoplasm, and as a consequence, the reaction is going to go more quickly with less energy wasted along the way. It's become more efficient. All right, easy peasy? Well, there's a little bit more involved than that. Here's the thing, folks. Not every reaction that our cells carry out is a thing we necessarily want to have happening all the time. Sometimes we are going to want to control that reaction and make sure it only happens when we want it to. There are two basic ways of doing this. One of them is through using a molecule called an activator. An activator is a small regulatory protein that binds to the enzyme. Think of the activator as being like the key in the ignition of your car. You put the key in the ignition, the car turns on. The activator is turning on the enzyme, making it into a condition where it can actively carry out the reaction that it needs to. The way an activator works is that the enzyme that needs one is an enzyme that you might say is turned off by default. That enzyme is not active under normal circumstances. The reason it's not active is because its active sites do not match the shape of the substrates, that by default, the active sites are flexed into an inactive configuration, where the substrate can't bind to them. When you put in the activator is going to go to that enzyme and bind onto its allosteric site. Remember we said we have the allosteric sites where helper molecules can bind on. This is one example of that. The activator binds to the allosteric site of the enzyme. When it does, it causes the active sites to change shape. They flex into a shape that now matches the shape of the substrates. Now the substrates can go in and do their thing. As long as that activator is in place, it's like having the key in the ignition of your car, that enzyme will remain active and it will keep on being able to carry out that reaction. Take away the activator and the enzyme shuts down. The substrates can no longer bind because the activation sites have now flexed back into their inactive configuration. So activator is there, enzyme is turned on, active sites match the substrate. Take away the activator, enzyme turns off, active sites no longer match the shape of the substrates. In this way, we can use activators to turn on enzymes only when a desired kind of reaction is wanted. Let's go the other way. Maybe we have an enzyme that instead of being active only when told, maybe we have an enzyme that is active by default, an enzyme whose normal condition is to be in an active state. For an enzyme like that, we want the ability to turn it off when needed. It's an enzyme that is on until told otherwise. So we don't need an activator in this case. What we need is an inhibitor. I-N-H-I-B-I-T-O-R, not E-R, inhibitor. My inhibitor is a molecule that deactivates the enzyme, preventing it from carrying out the reaction that it would otherwise carry out by default. No enzyme needs both. You're either an enzyme with an activator or an enzyme with an inhibitor. Inhibitors can work in two basic ways. You can have what are called active or competitive inhibitors. A competitive inhibitor blocks the reaction by getting in the way of the substrate. The competitive inhibitor acts like a cap over the activation sites, covers up the activation sites so the substrate can't get in. That inhibitor is in the way, blocking off access. And as long as it stays there, the substrate is unable to bind to the enzyme. The reaction stops. Take away the competitive inhibitor, and the enzyme will be able to get in. But as long as it's present, it's capping off the active sites, and the enzyme is shut off. The other way inhibition can work is through a non-competitive inhibitor, also called an allosteric inhibitor. My non-competitive inhibitor does not physically get in the way of the substrate. As its name suggests, it works by binding to the allosteric sites on the enzyme. It's like the opposite of an activator. If you think of the activator as being like the key in your car's ignition, you might think of the inhibitor, the allosteric inhibitor, as being like a lock on a door. You put the inhibitor in place, and the enzyme flexes into an inactive shape. It's the opposite of what the activator is. As long as that inhibitor is bound to the allosteric site, the enzyme will stay in a shape where its active sites don't match the substrates. Take away the inhibitor, the active sites will flex back into a functional condition, and the enzyme will resume its activity. So competitive inhibitors physically get in the way of the substrate. Non-competitive or allosteric inhibitors bind to the enzyme and change the shape of the active sites so that they no longer match the substrate. Either way, take away the inhibitor, the enzyme resumes activity, it's turned on by default. There's a nifty way that inhibition can work that we call feedback inhibition. In feedback inhibition, you're going to have a situation where the product of the enzyme's reaction is going to be the inhibitor for that enzyme. In other words, whatever thing that enzyme is helping to make is also its inhibitor. Here's the way it works. Let's just suppose you've got a cell that needs a certain concentration of a given ion or given molecule. And we've got an enzyme that makes that molecule. So the enzyme begins churning out that molecule, carrying out that reaction, and as we make it, the cell begins using it for whatever it needs it for. But let's suppose the level of that molecule is climbing and climbing and climbing, and it hits the point when the molecule now has enough. The cell has enough. The cell has the level of that molecule that it needs to do what it has to do with it. At that point, any more that we make, if the enzyme keeps on churning it out, is extra. And those extra molecules can now act to inhibit the enzyme because they're not being used by the cell for something else. They can be used to block off the active sites as competitive inhibitors, or they can bind to the enzyme as allosteric inhibitors. Either way, we call it feedback inhibition because the thing that the enzyme is making is also the thing that once you have enough of it will shut it down. The cool thing about that is that once the level of that substance falls below the needed level, there will no longer be enough of it extra available to inhibit the enzyme. At that point, the enzyme will turn back on and begin making more of that material. So you'll find you go back and forth. You've got a state of negative feedback where if the enzyme has if the cell has too little of something, the enzyme activates and turns back on to make more of it. If the cell gets enough of that thing inhibits the enzyme, stops making more until the levels fall again. So feedback inhibition allows an enzyme to be sort of self-regulating. It will stop making whatever product it produces when there's enough of it and resume making it when there's no longer enough. Make sense? All right. Now, this is all, standard enzyme operating procedure. This is all okay. Enzyme can either catalyze anabolic or catabolic reactions. We can turn it on with an activator in some cases. We can turn it off with an inhibitor in some cases. There are some enzymes that don't have activators or inhibitors. There are a few enzymes that are pretty much just active all the time. They never stop doing their thing. They're ready to go at any given moment. The enzymes that make ATP, for example, are constantly active. We don't activate them or inhibit them because their function is always needed. So not every enzyme has an activator or an inhibitor. Some of them don't need either one, but any given enzyme will only use one or the other. Now, here's the thing though. A lot of the chemistry that goes on in your body is going to be relatively complex chemistry. A single reaction is not always enough. Okay, so I have an enzyme that helps me combine A and B to make C. That's cool, but it may not be enough to carry out the task we need to. What if we're doing something super complex? What if we're doing something like aerobic respiration, in which you've got a multi-step process of metabolizing glucose and slowly breaking its chemical bonds to make ATP? Or what if we have a very complex chemical process like the synthesis of DNA, which requires linking together literally billions of small monomers to make that complex DNA chain? A complex multi-step chemical reaction like that cannot be done by one enzyme, because a single enzyme is not going to be able to carry out all the steps involved. A lot of our body's chemistry is not individual reactions. It is actually a series of reactions, one reaction feeding into the next one, and all of them needed to carry out the task we want to perform. We call that a biochemical pathway. So a biochemical pathway is a series of chemical reactions with one desired outcome. My biochemical pathway is a situation in which the product of one reaction becomes a reactant in the next one. Here I've got a sort of hypothetical biochemical pathway there. You see I'm combining A and B to make C. C then becomes the reactant for step 2 of the pathway, a second reaction altogether, in which C combines the D to make E. Then E goes on to the third and final step of the pathway, where it combines with F in the third reaction to make G. G was what we wanted the whole time. That thing at the very end of the pathway was the goal. But to get to the goal, we had to carry out not one, but 3 separate reactions. And therefore, this is our biochemical pathway. In this case, it's a 3-step pathway. Aerobic respiration is much more complex. It's more than 50 steps. The making of DNA requires more than 100 steps. There's a lot involved in some of the very complex pathways your body engages in. But the thing to remember about a pathway, it's a multi-step reaction, several distinct reactions as part of the path. The product of one reaction goes on to become a reactant in the next. Some of them get way more complex than what I'm showing you here. This is just to demonstrate the point. Now, key thing about this, remember folks, there are no all-purpose enzymes. Any given enzyme can only carry out one particular kind of reaction because it only binds one particular kind of substrate. It's got active sites that match the substrates for that one reaction. Other substrates won't fit. So if I have a pathway like this, a 1, 2, 3-step biochemical pathway, I need 3 separate enzymes to do that. I'm going to need an enzyme to carry out step 1, and a totally different enzyme to carry out step 2, and a totally different enzyme to carry out step 3. Because the enzyme with active sites for A and B is not going to match C and D or E and F. In other words, I need 3 different reactions involved here. I need 3 enzymes to do the job. So biochemical pathways typically require a series of enzymes, one after another, all working together to get the final goal to happen. A short pathway might just be 2 reactions, 2 different enzymes. A long one might require dozens. Now, clearly, we've got an issue here where we're passing things from one enzyme to the next in our pathway. We want to keep this going efficiently. We want to make sure that the product gets from one enzyme to the next in as efficient a way as possible. We want to make sure that we don't lose anything along the way because that would disrupt the whole process. So let's consider what happens if we disrupt it. Maybe we can't finish our pathway. Maybe if you're missing an enzyme, the pathway can get started, but it can't be completed. And at that point, the whole process breaks down. Fun example. So-called Indian corn that you get in fall, right? You know how it's got all those different colored kernels. You've got the really dark bluish-black kernels. You've got the red kernels. You've got the white kernels. What's happening there is it's actually a 2-step biochemical pathway. The pathway involves breaking down pigments within the kernels of corn. You need 2 enzymes to break down the pigment completely. If you are missing the first enzyme in the pathway, then you can't start the pathway at all. The pigment is not broken down, period, and the kernels stay a nice dark blue-black color. If you have the first enzyme in the pathway, but not the second one, you can take part of the reaction, you can partially break down the pigment, but you can't completely break it down. The partial reaction causes it to turn red. So when you see the red kernels in Indian corn, those are the ones that only had one of the 2 enzymes needed for the pathway. They could start it, but they couldn't finish it. The white kernels are the ones that had both enzymes that were required. Those enzymes, both are present. You can finish the 2-step pathway to break down the pigment completely, and the kernel therefore turns white. Now, to keep things running efficiently, to make sure that our pathway goes the way that it is supposed to go, there are a couple of ways that the cell can assist with this. One of them is through the use of what we call a multi-enzyme complex. A multi-enzyme complex is a cluster of enzymes, all the different enzymes needed for that pathway, clumped together. By keeping all of those enzymes in one place, we ensure that we can smoothly pass the product from one enzyme to the next. I finished it over here. I just pass it right to the next enzyme in line. It finishes it. It passes it right to the next enzyme in line, and so forth. By keeping all the enzymes together, the multi-enzyme complex ensures efficiency. It cuts out the risk of losing the product along the way or having a delay of it getting from one enzyme to the next smoothly. By just passing it along, we keep that pathway going as fast as we can. The multi-enzyme complex might be a temporary structure that is only formed when the reaction is needed, or it might be a permanent structure, almost like a little organelle that's always present in the cell, ready to carry out that reaction. Another way of making things efficient along the way here is with the help of a molecule called a coenzyme. A coenzyme is usually a small organic molecule, typically a vitamin. Vitamins are often coenzymes. That's what they're for. And what the coenzyme does is that it acts like a shuttle, carrying things from one step of a biochemical pathway to the next. The coenzyme picks up the product from enzyme 1, shuttles it to enzyme 2. Goes back, picks up more, does it over again. So by having that coenzyme at work, we can continuously move the products between enzymes and help make sure that that reaction keeps on going smoothly. We're going to talk about some very important coenzymes in the near future. Whenever we talk about respiration next week, we're going to see that a couple of coenzymes, most notably one called NAD, are vital to the process of aerobic respiration. You can't make ATP without the help of those coenzymes, shuttling molecules between one step of the respiration process and the next. So that's another way that we can speed things along. Now, a consideration here with a biochemical pathway that's kind of parallel to the enzyme thing, but it's worth discussing, is that ultimately, when you're carrying out lots and lots of reactions at once, you're probably breaking a bunch of chemical bonds. And when you break a bunch of chemical bonds, you're releasing a lot of electrons. Those have to end up somewhere. Free electrons flying around, free radicals, you've all heard that term. Free electrons flying around can set off other chemical reactions that we don't want to be happening. So we want to do something with them. We want to soak them up. All those excess electrons being cut loose from our pathway, we want to deal with them somehow. For that purpose, we're going to need what's called a cofactor. Cofactors are associated with a lot of biochemical pathways. They are usually inorganic minerals. The minerals in your diet, like zinc and so forth, are often going to behave as cofactors. The cofactor doesn't help the enzyme out directly. Rather, what a cofactor is doing is it's soaking up excess electrons that are released by a biochemical pathway. That ensures that those electrons aren't setting off other unwanted reactions as part of the cell, and thereby makes the whole thing work a bit more efficiently. One last thing I wanted to mention before we tie off enzymes, folks, and that is the fact that there are a very few enzymes that aren't proteins. By default, most enzymes are proteins. In fact, if you see a textbook say enzymes are a type of protein, they're not wrong. But we have discovered in recent years that there are a very few enzymes that instead of being made of protein are made out of RNA. They're called ribozymes, R-I-B-O-Z-Y-M-E-S, ribozymes. Ribozymes are going to be RNA-based enzymes. They help with a lot of chemical reactions that are involved in the manufacture of DNA, very basic fundamental processes that need to be carried out by the cell. You're going to find that ribozymes are very, very important to a conversation we're going to have at the beginning of Bio 152, the question of how exactly did life come about in the first place? Because one of the big problems with the origin of life is how do you build protein enzymes without DNA, but how do you build DNA without the help of protein enzymes? And the answer to that question is you don't. Use RNA-based enzymes, ribozymes, to carry out that critical first step. So we don't talk about ribozymes all that much. There aren't many of them in our cells. They're only carrying out a very limited range of tasks, but those tasks are pretty important. All right, folks. So that is my lesson on energy and enzymes wrapped up. That's all the stuff that I want you guys to know about that. Obviously, make sure you do the reading in the textbook on this topic. You know, there might be some details in there that I didn't throw into this. But you've now heard the things I expect you to know about energy and enzymes. We're done with this chapter, and by extension, we're done with Unit 2. We're ready to move on to new things. So here's the deal. On Blackboard, you will find the assignment on cell structure. Make sure that you have that completed and ready to give to me on Monday whenever I come back from my travels, unless you've already handed it in. Make sure you have all your lab assignments, as I've given you in your lab section, ready to hand in to me that day as well. And make sure you go on Blackboard and look for the study guide for Exam 2. It might be listed as Midterm Study Guide or Exam 2 Study Guide. I'm not sure which one. Either way, it's posted. It's available for you. Start looking that over, because when I come back, we're going to be talking about Exam 2 on that first Monday that I'm returning to class. I'm probably going to be assigning it within a day or two. It'll be a take-home exam, but you want to get ready for it as soon as you can. All right? That's all I have to say to you. I hope you all have a great weekend, and I will see you on Monday the 18th. Bye.